All right, let's finish up these final pages of the PowerPoint. So we want to talk about what is the difference between a vacuole and a vesicle. What is the difference between a vacuole and a vesicle? Well, vacuoles and vesicles, we know are membrane packages. We've seen them before when we talked about the endoplasmic reticulum, for example, pinching off and forming this membrane package called a vesicle that would either travel to other parts of the cell or travel to the plasma membrane to deposit the contents of the vesicle to the outside of the cell. So vesicles and vacuoles are membrane packages, and it says they are unwrapped by Golgi. We know um, if something goes to Golgi and unwraps it, modifies it, repackages it in a vesicle, and that vesicle again, coming from Golgi, would move either to the plasma membrane for exocytosis or to some other parts of the cell. So the main difference between the two is going to be the size. Vacuoles are generally larger than your vesicles and the contents that they contain inside. So vacuoles, we say, are large membrane packages, and generally they house solid materials. So generally they would house it said solids. Okay. And then the vesicles are small membrane packages, and we say that they generally house liquids. Okay. But there are exceptions to the rule. We can have a vacuole, for example, like the large central vacuole, which is found in plant cells, and it has liquid on the inside. Water, sugar, salt, all this stuff that's in solution inside of that large central vacuole. And then we could have a contractile vacuole that pumps excess water from cells. All right, so let's move on to the microtubules versus microfilaments, and then I also will probably mention intermediate filaments. These are all the filaments that you'd find inside of a eukaryotic cell. All right, so it says these are protein threads found inside of the cytoplasm. Microtubules are hollow threads made of tubulin. So microtubules are hollow threads made of tubulin. And again, for example, if you took a hose pipe and looked down the center, we know that it's hollow. So that's what we're talking about. It's hollow. It has an open center. Um, one of the things that we find out about microtubules as far as some of their function, it says, and I put a video link in here, I think, so that you could actually see what's happening inside of your cells. But anyway, these guys are part of two classes of motor proteins, and namely the kinesins and the dyneins. And the kinesins, it says they move along the microtubules, usually carrying cargo such as organelles or vesicles. Vesicles. Now, one of the things that you'll see, and even if I didn't put a video in here, you can Google kinesins and dyneins, and the difference between the two is one is moving in one direction, one actually is moving cargo in toward the interior of the cell, the other one's moving cargo towards the exterior, okay, of the cell. So that's the only difference between those two types of microtubule tracts. But anyway, then the microfilaments, we know that these are the thinner filaments. These are made of actin. If you've ever been in a anatomy and physiology class, then you probably have heard the word actin. You probably heard the word myosin. These two guys work together as far as making sure a muscle contraction takes place. The myosin is the thicker of the two, and it normally has two heads that attach and have this ratcheting effects so that it shortens a particular muscle. But anyway, the intermediate filaments, threads, also another protein filament. But anyway, all three of these make up your cytoskeleton. And the cytoskeleton, again, a network of tubules and filaments that extend from the nucleus all the way to the plasma membrane. And here's a picture of that. And again, we said, here's your plasma membrane all the way out here. Here's the nucleus. So these filaments, they stretch all the way from the plasma membrane over to the nucleus. And you can see the various organelles that are suspended inside of the cell. And guys, I apologize for my voice, but I am extremely hoarse, probably from exhaustion, but it's gonna be all right. Again, mention the actin filaments. If you're going to have a muscle contraction, most likely you're gonna have the actin filaments present in that cell. Generally, there's two chains of the actin filaments that are twisted in a helical manner. And we know that muscle cells have many of these actin filaments, and that's not the only protein thread that you're going to find in or even involved in a muscle contraction, because remember I mentioned myosin, another protein molecule that's present, and even more than that. So and you don't have to know this particularly, this is just giving you a little background information. All right, so the microtubules, this you definitely need to know. The microtubules, they make up what we call centrioles. Usually you're going to find two centrioles per cell, and then during the division process, because centrioles, what we find out, are responsible for helping with the organizing of spindle fibers. If you've ever had a biology class, you know that spindle fibers help to move chromosomes. So they make spindle fibers again in order to move chromosomes. Again, there's always two bundles per cell. They have what's called a 9+0 arrangement of those microtubules, and I'll show you a picture of this. If I drew a picture of this, it would look more like this. This is my rendition of the two microtubules, or the bundles of microtubules, but I will show you what the real one looks like. So again, these guys make up, there we go, the MTOC, which is the microtubule organizing center for the cell. Okay, let's move on. More movements, because all these filaments that they're talking about, we said that they are responsible for movements that are taking place in the cell. So two others, cilia and flagella, the organelles which move cells by a paddling or whip-like motion. The cilia, we know, are the short-haired structures. And if you remember, we've all had this description of short hair-like structures before when we talked about the fimbriae or pili that were found on bacteria cells. But the difference between the fimbriae or pili, which are also referred to as short hair-like structures, is that the fimbriae and the pili on the bacteria help them to attach to surfaces, whereas cilia and the cilia is going to be for movement. Okay, so places that we find in unicellular paramecium, we also find these guys lining our respiratory tract. So and so the function is to help this debris that we're breathing, the dust and so on and so forth. It says it catches that And then when we're coughing, that mucus kind of traps it as well, and we're able to move it back up and hopefully spit it out. A lot of people you see creating those wonderful, nasty, what young people call loogies. Anyway, all right, so we said they line the cells of the upper respiratory tract. Now the flagella, these are long whip-like structures, and for example, we'll find them on the sperm, help to propel the sperm forward so that he hopefully make it up through the vagina, into the uterus, and then up to the fallopian tube in order to fertilize an egg. Okay, so both are composed of the 9 pairs of microtubules. So they have what's called a 9+2 arrangement because there's 2 of the microtubules running down the center. So it's not hollow like what we saw on the previous slide. And I'm going to back up to the side. See, there's nothing running down the center. So it's a 9+0 arrangement of microtubules. So 9+0. There is nothing in here. It's hollow. Okay, so let's go back to this slide where we've got this 9+2 arrangement of microtubules. So, and it's when the doublets pass each other that you have the whip-like motion. And let me get that picture up here. So here's the 2 in the center, and then you have a doublet here, a doublet there, a doublet there. Okay? And I believe this concludes our slides for this chapter. Now, I will not cover the chloroplast and the mitochondria until we get to the chapters that they are, well, I guess I could briefly mention them, but we won't get into the vast detail until we get to the chapters that involve them. So photosynthesis will be the chapter that we'll use to talk about the chloroplast, and cell respiration will be the chapter that we use to talk about the mitochondria. Okay, so mitochondria, pill-like structure, and chloroplast also pill-like structure, but mitochondria we know are known as the powerhouses of the cell, and then the chloroplasts, we know that their job is to carry on photosynthesis. So they harvest the sunlight, utilize some water and some carbon dioxide, and make us some wonderful sugar and oxygen. All right, so I will see you next time. Hopefully my voice would have cleared up by then.